The marine embryonic stem cells are stored in liquid nitrogen. They are divided into portions of 5 million cells each in cryogenic vials and must be thawed first. Therefore, the removed tube is rapidly transferred into a water bath at 37 degrees Celsius. Then, the stem cell suspension from the cryotubes is transferred into a centrifuge tube. Remaining cells are rinsed with 2 times 1 milliliter of culture medium and also transferred into the centrifuge tube. The tube is centrifuged for 5 minutes at 900 G. Meanwhile, a culture flask with 5 ml is filled with LIF supplemented culture medium. LIF stands for leukemia inhibitor factor. It prevents the differentiation of the cells at this time. The supernatant containing the freezing medium is discarded and the cell pellet is resuspended with additional 5 ml of culture medium with LIF and is also transferred into the culture flask. The culture flask is incubated for 24 hours at 37 degrees Celsius and 5% carbon dioxide, following a further change of culture medium. After further 24 hours, the cells are passaged. To this end, the exhausted medium is first vacuumed off. As the cells adhere to the bottom of the culture flask, they stay in the bottle. Dead cells are rinsed off with 10 ml of PBS. To release the cells, 1.5 ml TRIP-LE Express are added, which is a trypsin substitute. The solution is left in the flask for about 90 seconds until all cells have detached. Remaining cells can be removed by gently shaking and tapping. The reaction is stopped by adding 5 ml of culture medium. The cell suspension obtained is transferred into a centrifuge tube and centrifuged. The supernatant is discarded and the pellet is resuspended with about 5 ml of culture medium to relieve the cells from TRIP-LE. Part of the cell suspension is removed and treated with trypan blue. Then 10 microliter of the suspension are added to a Neubauer chamber and counted under a microscope. For further disposition, a new culture flask is filled with 10 ml of culture medium with LIF. Then 5 million cells of the cell suspension are added. The flask is incubated for 48 hours at 37 degrees Celsius and 5% carbon dioxide. With the remaining cell suspension, the further testing can be performed. For the proliferation assay, 50 microliter of 1% gelatin are added each in the wells of a 96 well plate. The gelatin enhances adherence of cells to the bottom of the wells. The plate is incubated for 2 hours. A small petri dish is filled with 10 ml test medium. The cell suspension previously created is diluted to a target concentration of 10,000 cells per milliliter. "50 microliter of this cell suspension is transferred into the wells via a multi-channel pipette. It should be ensured that the outer wells of the plate remain free. For adherence of the cells, the plate is incubated for 2 hours again. Then 150 microliter of test medium are transferred into the wells. 1 test concentration is used per column. The outer wells of the plate are filled with PBS. The buffer prevents drying out of the inner wells after being placed in the incubator. After 72 hours, a medium change is performed. The exhausted medium is vacuumed off and replaced with an equal amount of fresh medium. After another 72 hours in the incubator, this step is repeated. On day 10, the plates are freed of the test suspension by inversion. Since the further timings are short, sterile work is no longer necessary from this time on. The remaining cells are washed with 100 microliter PBS per well. Subsequently, the buffer is discarded by inversion of the plates. "100 microliter of preheated MTT solution are added in each well for the detection of cell viability. The yellow dye is reduced by the cells to purple formazan. After 2 hours of incubation, the plates are centrifuged for 3 minutes at 2500 G. The supernatant is then discarded by inversion. In the final step, 100 microliter DMSO per well are added. DMSO dissolves crystals of the formazine dye, which could interfere with the measurement of the optical density. The plates are placed into the photometer and shaken there without light irradiation for 15 minutes. Subsequently, the optical density is measured at 570 nanometers. For the differentiation assay, a cell concentration of 37,500 cells per milliliter is needed. First, 5 ml of test medium are given into a small petri dish. Accordingly, 185,000 cells from the previously prepared cell suspension are added. The bottom of large petri dishes is covered with sterile water or PBS. This serves as a moisture buffer and prevents drying out of cells in the incubator. Subsequently, 20 microliter of cell suspension are applied to the lid of the dishes with a multi-channel pipette in the form of drops. One drop contains about 750 cells. The drops should be spaced closely together to be able to accommodate as many as possible. The lid is placed on the tray and is incubated for 72 hours. By gravity, the cells form cell clusters at the bottom of the drops, so-called embryoid bodies. With 5 ml test medium, the embryoid bodies are rinsed off the lids. It should be ensured that no embryoid bodies are left behind. They are transferred into bacteriological petri dishes and incubated in these for 72 hours. During this time, the embryoid bodies grow and significantly increase in size. In a 24 well plate, 1 ml test medium is added per well. Then, exactly one embryoid body is added to each of these wells. During an incubation period of 96 hours, the embryoid bodies can grow at the bottom of the wells and further differentiate into cardiomyocytes. This differentiation is taken as test endpoint. Therefore, one observes the contraction of the cells. Wells with contracting cells are counted.